Hello and welcome to this brief video on RNA. RNA is the slightly more simplified relative of DNA and acts as a sort of bridge between your genome and proteins, the control module of enzymes, and more. Despite this important activity and other more recent findings, it does not get as much attention as DNA. RNA is arguably the precursor to DNA and is a simplified version of it. Under normal circumstances, it exists as a single strand of nucleic acids that turn back on itself to remain intact. Unlike DNA, it is made up of guanine, uracil, adenine, and cytosine. In this case, uracil replaces thymine that would be found in DNA. Unlike DNA, RNA has a ribose, not deoxyribose backbone. This makes RNA far less stable than its DNA counterpart. RNA is made through a process called transcription. That is taking the DNA helix, opening it, and reading the gene that is being transcribed. Depending on what purpose the RNA has been transcribed for, this can then be modified with additional features. An example of this is polyadenylation, which adds a whole series of adenosines to the end of the messenger RNA. This is called the poly-A tail. This is important in three of the major examples of the types of RNA. The first is messenger RNA. This is what leads to the creation of proteins. It consists of 3 RNA nucleotides, which correspond to an amino acid. The second is ribosomal RNA, which is part of the ribosome subunit, which creates the whole complex in which RNA produces proteins. The third is transfer RNA, which acts as a chaperone molecule for the amino acids, which bind with the other amino acids to create a protein. As you can see from this application, RNA is ubiquitous within life. It is also seen in the questionable area of life that is viruses. Ebola is a case of this, wherein the virus responsible for Ebola is made of a single strand of RNA. Other than these expression-related RNA molecules, there are also those which perform a regulatory function, pieces of RNA that can interfere with expression, transcription, or even access to certain genes. The interference occurs through several types of RNA. This largely includes the 98% of the genome, which is not responsible for expressing a product. It includes RNA like microRNA, which are small molecules, which is self-evident in their name. What is less obvious is how they interfere with messenger RNA by binding with a corresponding sequence, which prevents the unit from working with ribosomes. Without messenger RNA, there is no instruction for the ribosome to produce protein. Then there are long non-coding RNAs, which are a new and emerging field of research. They are larger than microRNA, but still small and non-translating. It is thought that these are generated from some of the large areas of the human genome, which are not responsible for expressing protein or regulating genetic expression, the otherwise called junk DNA. This is supported by the regular reporting of involvement in transcription. A very significant area of study is cardiovascular disease, where it is thought that they are involved with the higher incidence of this condition amongst males than females. Like microRNA, enhancer RNA is small, but in this case it increases transcription by binding to a section of the genome and making it easier to translate. As with DNA methylation, these RNA modifications can work both ways by impeding or helping to facilitate gene expression. These various RNA molecules are very useful in molecular and cellular biology. In this case, RNA acts as a means of measuring the activity or even presence of certain active genes. This occurs through reverse transcription, which takes the RNA molecule, identifies its sequence, and then copies it repeatedly. Depending on how many copies are made, a PCR reaction will respond more or less strongly. This is then used to create a standard or reference measure of activity. One point worth mentioning from earlier is that not all RNA is single-stranded, but most is. There are examples of both circular RNA and double-stranded RNA. Double-stranded RNA is commonly seen in cells, bacteria, and especially viruses. Circular RNA is less common, and still something of a mystery. Overall, it was long thought that something like RNA was not necessarily responsible for the complexity of life. That was attributed to protein. Then we realized it was DNA, but still there were questions about how something so simple could create something as complex as life. Now we are seeing that the intermediary RNA is playing a major part in the role of life's complexity by helping to moderate, modify, and create essential products or facilitate gene function, on top of the already surprising complexity of DNA, which leads to something extremely complicated and extremely malleable, adaptable, and flexible to outside pressures. Thank you for watching this video. If you have found it interesting, please consider liking, sharing, and subscribing. If you have any comments, questions, or suggestions, please put them below.